Yes, we can see it.
Wonderful. Okay, thank you. Yes, hope you can all hear me. My name is Dr. Akbar Ganatra. I have a PhD in medical parasitology from ISIPE, and I'm going to be introducing essentially a strategy slash product that I believe can help eliminate schistosomiasis. And I call it the knock knock. So you can see I had a little play on words here. Knock knock, who's there? Schistosomiasis elimination. So this is what I'm going to be talking about today. Uh, is it working? Yeah. So a little bit about schistosomiasis. I think we all know that there's over 200 million people infected right now, and the fatality numbers range between 20 to 200,000 people per year. A bit about the life cycle. So if you have an infected person here, they're living with the adult worms inside their body, and these worms are basically mating and laying eggs that are being excreted with urine or stool. So when this urine or stool enters fresh water, for example, a river or a lake, the eggs hatch into a miracidia. To keep in mind that this miracidia has to search for a snail, and it can only do so within like 24 hours. Otherwise, it just loses energy and dies. But a single miracidia infecting a single snail basically releases thousands of cercari. They can release hundreds of cercari a day. So the miracidia infects the snail. They undergo asexual reproduction, and then this leads to releasing human infecting cercari, which you can see also pictured here. These also have about 24 to 48 hours of energy before they can seek a human. And if they do infect a human, this stage needs two of the larvae to infect a human to continue the life cycle, because they need to be paired in male and female. But again, I put this simplified version because the main point I want to drive home is there's these two bottlenecks in the life cycle where we want to break the egg and the cercari. So because these both have about 24 to 48 hours to live, and I feel like these are obvious targets for any interventions. A little bit about my experience, my PhD experience. So I was in Western Kenya, and this is near Lake Victoria. This is a map of the endemicity and the prevalence of schistosomiasis in the area. You can see it's scattered quite highly, and in some areas it's 50 to 100%, quite high. So yeah, I scattered my points just like that, and I went collecting snails and testing humans as well. But here I only have pictures of me collecting snails and other insects. A little bit of the side information about my PhD. It was the effect of pesticides on transmission of schistosomiasis. Essentially, what I found was when the insecticides you put on farms enter the water, they kill the insects, which are usually competitors or predators of these snails. So the number of snails increases, and so does the risk. However, I'm not here to talk about that so much. I just basically wanted to go show you more examples. This is essentially a river sow that is in Western Kenya, which I just showed you. It was one of our most snail infected and schistosoma infected sites. So I don't know if I can have a laser. This, I can't see the option right now. But yeah, this bottom right site was where we found the most snails. So in my curiosity to find out what kind of sites do the snails prefer, I went down this river. This is a 16-kilometer stretch down the river, and I was searching for snails every 500 meters. And essentially, we didn't find that many snails, me and a master student. But what we did see is that the community essentially allocated certain parts of the river for different things. So you can see this woman is here cleaning her dishes. If you go a little bit further down, this man is here cleaning the motorcycle. And then even further down, there's where the animals come to, where the people bring their livestock to feed and drink and stuff. And of course, in between, there are male washing areas, female washing areas, which for obvious reasons I haven't shown pictures of. But for me, this really struck that the points of contact with the rivers, with the infected waters, are very localized. The people naturally only go to the parts of the rivers that are safe enough. And even in those, they have designated areas so that they can carry out all their domestic functions with the water, together, cohabiting. Yeah, so those are my observations from the field, and obviously I was researching schistosomiasis for about 6 years. I went to see what is the current advice given to defeat transmission, and this is from WHO or CDC. Essentially, what they say is, well, avoid water that might be contaminated with schistosomiasis. It's a little bit easy to tell a traveler that you maybe if you're going to Kenya or another country that you're not familiar, the tropical country, maybe don't swim in a lake or a river. But for the people who are living in the area, this is the water, like you said, that's what they use for all domestic uses. You can't really tell them not to touch the water. So the other advice is either boil your water or store it for 48 hours. This is supposed to be for 1 minute, sorry, not 3 minutes. And yeah, essentially don't touch the water. So I summarized all this as, well, we should try preventing contact with of people with the infected water. So I put this as no contact. The next thing is, of course, well, humans are also infecting the snails through the urine and the feces or the eggs being released with those. So the second bit of advice is try to limit that. So essentially use clean toilets and things like that. Again, this is very, much traveler advice. So we're seeking for a solution for the people who actually live in these areas. But the point of this is you should not contaminate. So I put this as no contaminate. You will try to limit the number of eggs entering the system. And the final one is, well, essentially, it's a neglected disease. No one talks about it. And even though I might come off a little bit naive in believing that we can eliminate it, I do believe we can eliminate it. And one of the only ways to do so is to talk about it and really get the discussion going. So I put this one as educate. So how does it all come together? So I envision something like this, a knock-knock wash point. So the first thing is it's targeted. We go to the parts of the river where people are already using that river for certain things. Over here, I have a urinal/shower. I hope it comes off. I know the graphic is not exactly looking very well, but it's supposed to be purpose-built for the activity happening at each site. So over here, I've assumed that this is a washing area. Um, since behavior changes is one of the biggest problems we have, we are bringing the solution right where people are using it. It's like a grassroots solution. So we bring the wash point here where people are using it. We take the same water that people are used to using it. We use a very cheap intervention, which is just a reservoir that holds the water for 48 hours. And this, I think, can also help in, reducing the distrust because people can see that there's nothing being added to the water. Of course, there are more other interventions. You can use UV, you can use chemical, you can use sand filters, but just for simplicity, I put a reservoir here. So this satisfies the no contact. We were taking the water from this river and we're dispensing it cleanly and free of sacchari, essentially. Hopefully governments can take it up and provide, control for pollutants and other things like that. But for schistosomiasis, all we care about is that it's free of sacchari. The other part, like I said, this is a urinal. It doesn't look like one, but we've also tried to partner with Mary Lou. And again, the point of this is it should be a bathroom. It should be placed where the people are going to, defecate near these rivers. And, we can deny it happens, but I've been there and I've seen it and it does happen. So rather than guilt people or shame people, I think we can provide the facilities for them. These are the Mary Lou is like a above ground pit latrine, which really digests all the fecal matter. And within a few stages, you can actually water your plants with it. And yeah, I also envision lastly that you can have it in a way that teaches the people about it. So yeah, just to conclude, mass drug administration right now is the only tool used, but it does not prevent reinfection. And we currently say that, yeah, we need to break the life cycle. Like I said, there's two short-lived life stages we can target. WASH is hailed as a necessary complementary, but we don't really specify what that means. And this is what I'm trying to do. We specify no contact, no contaminate, educate. Yeah, most WASH interventions also don't take cultural differences into account, which this does with the WASH point. And finally, yeah, knock knock, I think is quite a catchy slogan we can use to try spread the elimination message. Sorry for going over time. Thank you so much.
Well, thank you for the beautiful presentation. Thank you for the acronym. Is the acronym or new word?
Yeah, knock
Is it
knock.
is interesting. Does anybody have any question to ask? Before anyone asks, let me ask you this question. How visible is the low contact component in communities where their livelihood depend heavily on daily water contact, such as
Yeah,
fishing or
yeah,
farming?
yes. Right. So there are challenges. I would say that firstly, my experiences was really based on the inland waters of Western Kenya. So not really in the lake, but the rivers that feed into the lake. So these
Okay.
communities, as you can see, if you remember the map early on, they are also quite heavily infected because there's a schistosoma in the biomphalaria. There's Sudanica, which lives in the rivers. I mean, Faferi that lives in the rivers, where Sudanica and Kwamfala live in the lake. So this can address the rivers for the people, but it is kind of modifiable. It like for the place where the people are bringing the livestock, you would maybe have an animal trough. For the places where people are showering, you would have like a shower or something where they're washing their clothes, a place for the women to sit. So one of the important parts I didn't really mention is you would have stakeholder events to say, how are you using this water? How can we make it safe for you? We want to ensure, we let the people know, we want to ensure that if you want to collect this water, you don't put your feet inside. You can get it dispensed to you. And I would say for fishermen, as long as you keep to the tenet, which is no contact, no contaminate. It's a bit difficult with the contact stuff, but I think you can definitely prevent contamination. Maybe, like I know the people openly defecate into the lake when they're fishing for long periods. I think that is maybe can be solved with a bucket on your fishing boat or something like that. I mean,
Okay.
it's not impossible. Yeah.
Okay. Thank you very much. Okay, I think
No, no
we
problem.
can. Thank you very much. I think we can move to the next presentation. Uh, Um, Ibikunle. Okay, you're right. Let me share your the PowerPoint.
Can I start?
Oh, let me. Sorry. Okay, I think you can start.
Good afternoon. My name is Ibikunle Elizabeth Oluwatoyin, and I'm representing on the topic prevalence and intensity of Rogenta schistosomiasis among school children in Spirwell community of Ikere Georgia, Iseyin, LGA, Oyo State, Nigeria. Schistosomiasis is caused by infection with blood flow of genus Schistosomia. Schistosomiasis infects more than 230 to 250 million people annually. Okay, my first slide is just talking about the distribution of schistosomiasis, the transmission, and how it affects the people in the world, the global people and the local people. And schistosomiasis has been controlled chiefly through mass drug administration of praziquantel. Now, the purpose of my study, the aim of this study is to determine the frequency of infection following mass drug administration and to identify the risk factors associated with congenital schistosomiasis in school children in the study area. Now I move to my methodology. Cross-sectional surveys on behavioral characteristics was conducted. Questionnaires were administered. Then urine samples were collected 5 months after treatment of mass drug administration. According to my topic, it was prevalence and intensity of congenital schistosomiasis. Then it was after mass drug administration that we went to the field to collect samples. So we went 5 months after the treatment. So urine samples are collected between hours of 2 a.m. to 2 p.m. Now, this was my data analysis. I used a chi-square to test the association of infection in respect to age and sex. Okay, so next slide. My results and discussion. Okay, I've been collecting samples from the field. I collected 109 samples, 109 samples, and I've been testing the for the egg of neurogenital schistosomiasis in the lab, and I also tested for hematuria. So we tested based on sex, based on age. So we used school age children, which are from age 2 to 16 years. So my first table here is talking about the result I got from the age 2 to 5, 6 to 10, 11 to 16. So when looking at it, the age 6 to 10, we are heavily infected with schistosomia hematobium. And 21 people were infected. Out of the 109 samples that were collected, 21 people, participants, sorry, they were infected with urogenital schistosomiasis. So my next slide, please. So this table is talking about the sex, male and female. Yeah, definitely male and female. Out of the 109, it was 21 participants were infected. So we have 8 male were infected and 13 female were infected. So overall, female had the highest prevalence and highest mean intensity. The next slide, please. This is the intensity of the infection. I've explained this, that female were more heavily infected. Okay, out of the 21, we found out that 2 particular females were heavily infected. That means the egg was 50, we had more than 50 eggs that were heavily infected. And so females were more infected compared to the male. Next slide, please. Hematuria, yeah. We had to test for hematuria in the lab using the combo strip. So we checked for blood in the urine. And looking at the blood in the urine, 15 were positive and 4 were negative. Yeah, out of the people that were tested for hematuria. Out of the 21 people, 15 were positive, 4 were negative. So the next slide, please. So I've been looking at my study, going to the field. This shows that there is still the prevalence in that community is low because out of 1 night we have 21 samples infected. This definitely shows that the prevalence, there's transmission, there's still ongoing transmission, but it is low at the moment. And low prevalence is likely due to past praziquantel FDA. Like I said earlier, according to my topic, there has been mass organization in the field before we went to collect our own samples, and this is one of the causes why there's low prevalence in that community. Females recorded higher prevalence and egg intensity, likely because they engage more in water contact activities, like female do the washing, do the bathing, do the washing of plates, washing of clothes, and because they use one source of water, one source of water to do majorly things of activities in the community, then there's more assurance that there will be transmission of infection. And children 6 to 10 are the highest proportion of infection, which is consistent with from reports for modern Nigeria and Africa story. Next slide. I think, okay, my conclusion. Urogenital schistosomiasis remain a public health concern in that community, Spirit community. Longitudinal studies are needed to investigate the snail intermediate host. I didn't work on snail host. I only collected urine samples, but according to the life cycle of schistosomiasis, we need to also deal with the snail intermediate host. Now, repeated MDA, that is the mass drug administration content, is recommended to eliminate persistent light infection. Since there's still ongoing transmission in this community, we still need to administer administer drugs to them content. Then the government should provide potable water because this transmission is majorly due to the fact that they use one source of water for cooking and doing washing and laundry. But if there's a borehole pipe on water where they can get clean water to cook, I think the transmission will reduce. Then improve sanitation facilities are needed to reduce dependence on contaminated river water. Thank you.
Thank you for the beautiful presentation. Is there any question? Does anybody have any question to ask? Ibikunle. Okay, there's not any question. I have some question for you. Okay, despite
Okay,
mass
sir.
drug administration, transmission is still ongoing for schistosomiasis. So, what factors
Okay.
do you think are mostly responsible for the infection in this community?
Okay, I think despite mass drug administration, they administered drugs previously and they're still ongoing. One of the thing I found out is that this um students, these participants, they're still engaged going to the river, the same source of river to wash their plates, to cook, to bathe. So there's the possibility that the transmission will still be ongoing. And most times, because after our work, we have to go back to administer drugs to them through the help of the state NLTDs, the health workers in the community rather. And sometimes there was one lady that was heavily infected. We didn't see her around. So this could be some of the causes again. They are not readily available. And most times, one of the major things I see is causing the issue is that they are still going back to the same source of water over and over again. So there is more awareness, more campaign. They need to know more so that they can avoid all these transmission issues. Thank you.
Okay, thank you very much.
Okay.
So, I think we should move to the next presentation. The next presentation is Epidemiology of Taeniasis by Ogudipe Olayinka. Ogudipe Olayinka, are you ready to present? Can you unmute?
Yes.
Okay.
Yes.
Should I share your screen or you share your screen from your end?
You can share. Very share.
Okay, I can share. Okay, I can share your. Okay, thank you. You can start. Thank you.
Okay. Good afternoon, everyone. I believe we can all hear me. Good afternoon, everyone.
Yes, we can hear you.
Okay, my name is Ogundipe Olayinka. I'll be presenting findings on a community-based study titled Epidemiology of Taeniasis in some community of Borno Demsa State local government area, Adamawa State, Nigeria. Okay, so just a little introduction of my subject. Taeniasis. Taeniasis is a human parasitic infection caused by tapeworm species. Taeniasis is a neglected zoonotic disease and can affect humans by eating raw or undercooked beef. That's Taenia stagnata or punk. Punk means Taenia solium or Taenia acetiaca, which for this food called
I think we can't hear you again, please. Can't still hear. We can't still hear you. No. Can anybody hear Olayinka? I can't hear him from my end.
No, there's no sound.
Okay. We can still hear you. Okay. Can we move to the next presenter then? When you fix it, we'll come back to you. If you agree, just type yes. Okay, let's move to the next presenter, then come back. Okay. I think I'm the next presenter. Next presenter. This is just a general. Just from the theme of the seminar. So we can just discuss generally on NTD. Like over 1 billion people, and they are mostly attributed to poverty, poor sanitation, limited healthcare access. So why do NTD persist? Sorry, Mr. Olayinka, we'll come back to you. After this presentation, we'll come back to you. Thank you. So why do NTD persist? We have different reasons why NTD persists. Health education is one key factor. Limited access to clean water, sanitation, inadequate health infrastructure, underfunding, and limited political priority. So from research to reality. For us to go from research to reality, we need to understand transmission. We need to understand the different life cycle of these parasites, of this organism. Then we can now understand how to break the
The sound has stopped.
the environment and also environmental interaction, because for a particular infection or parasitic disease to occur, all these factors need to come to place, especially parasite with vector. So evidence-based intervention based on the WHO aim of eliminating NTDs by 2030. We have the Mass Drug Administration, which is, we can see with ivermectin for lymphatic filariasis, where different health workers or public health scientists move throughout the community and give people this drug. And the evidence is showing, but not as the WHO planned. Because if you are trying to eliminate or eradicate NTD by 2030, at this point we should have like 60 or 70% of this disease eliminated. Then wash improvement. Yeah, wash is very key in eliminating NTD, because NTDs are attributed to sanitation mostly. health education and behavior change. Health education is very important. And yes, we have January 30 yearly to discuss, to sensitize, to carry out outreach, to inform people about this particular disease. But we need to move from the yearly sensitization to weekly, to daily, to even hourly for us to achieve the 2030 goal. The vector and intermediate host control, which are very key, because for the establishment of a particular parasitic disease, vector is very key, intermediate host is very key, especially with trematode like Schistosomiasis, Schistosoma, fasciola, they require intermediate host. So challenges in translating our research to impact, reinfection after treatment, and emerging diseases. For instance, the elimination of guinea worm was because the community were stopped. Elimination of guinea worm in Nigeria is because the community members were stopped from going into the river, and to stop them from going into the river, they had to give them pipe-borne water, and this actually prevents them going to the river. But if there is no pipe-borne water, the infection is going to continue, and with that, we can't eradicate or eliminate NTD. Drug resistance is a big concern with most of these parasitic disease, from schistosoma, fasciola, taeniasis, onchocerca, and the rest. Drug resistance is a big challenge. Funding, as we all know, and infrastructure, yeah, big challenge also. Socio-cultural barrier in which you need to educate, we need to educate the community, because a particular study that I read, I think last 2 years, where they carry out a survey and ask, what do you think cause NTDs? And most of them are attributing it to sham, to juju, and it's something. So we need education to break this social cultural barrier. And pathway to ending NTDs, multi-drug to sustain multi-drug administration alongside WASH, invest in research and innovation, strong political commitment, global and local partnership, and health education. So looking ahead, we need to move from control to elimination, and also empower endemic community, which is very important, because when we carry out our research, we get the results, we turn it into maybe it's a diagnostic kit or anything. The community needs to accept it and take charge. And to take charge, we need to educate them. And also, we should align NTD goals. We should align NTD goals with sustainable development. So to end this, I'll say research provide evidence. Community and policy turn this evidence into activities, and thereby we'll be able to end NTDs. Thank you very much. I don't know if there's any question. Any comments or addition to the presentation? Okay, if there's no comment, so Mr. Olayinka can present. Let me share your screen. Okay, I think you can.
Can you hear me now?
Hear you. Yes, I can hear you.
Okay, sir. Okay, good afternoon, everyone. I'm sorry again for the sound issue. My name is Ibikunle Elizabeth Oluwatoyin. I'll be presenting the findings of the community-based study titled Epidemiology of Taeniasis in some communities of Borong Densa local governments area of Adamawa State, Nigeria. A brief introduction. Taeniasis in human is a parasitic infection caused by tapeworm species. Taeniasis is a neglected zoonotic disease and can infect humans that eating raw and or undercooked beef, and that's Taenia saginata, or undercooked pumped Taenia solium or Taenia acetiata, which the Taenia acetiata is not predominant to my location at this time, so we will not be focusing much on that. Next slide. The statement of the problem. Taeniasis remains the next slide. Taeniasis remains a neglected public health problem, especially in the endemic region where my location is affected. And despite all the advances even to improve the epidemiology of taeniasis, taeniasis still remains prevalent in some regions, which my location is affected. Vulnerable populations in low and medium income countries also, and the control effort has been limited as a result of poor access to health care, sanitation, education, and then the asymptomatic nature of the taeniasis has also affected the control effort thus far. And so the aim of this objective, the next